Microbiology for 3rd semester Faculty of Medicine. 1. Fundamentals of microbiology. A definition of microbiology. Microbiology is the science that studies microorganisms, including bacteria, viruses, fungi, and parasites, as well as their interactions with the environment and other organisms. B. Classification of microorganisms. 1. Bacteria. Unicellular organisms that can be pathogenic, disease-causing, or non-pathogenic. Examples: Staphylococcus aureus, Escherichia coli. 2. Viruses. Non-cellular organisms that can only reproduce inside a host cell. Examples: Influenza virus, HIV. 3. Fungi. Eukaryotic organisms that can live as saprophytes or pathogens. Examples: Candida albicans, Aspergillus. 4. Parasites. Organisms that live in or on a host and benefit at the host's expense. Examples: Plasmodium, causes malaria. Entamoeba histolytica, causes amoebiasis. C. Basic techniques in microbiology. 1. Gram staining. A technique used to classify bacteria into 2 major groups: gram-positive, purple, and gram-negative, pink. 2. Microbe culturing. A technique used to grow microorganisms from samples using agar or liquid culture media. 3. Microscopy. Light microscopy is used to observe microorganisms in prepared slides. Electron microscopy is used to observe finer structures such as viruses. 2. Medical microbiology. A normal microbiota. 1. Definition. Normal microbiota refers to microorganisms that live in the human body without causing disease. 2. Locations. Found on the skin, gastrointestinal tract, upper respiratory tract, and urogenital tract. 3. Role. Normal microbiota plays a protective role against pathogens by competing for living space and resources. B. Pathogens and diseases caused by microorganisms. 1. Pathogenic bacteria. Escherichia coli, causes diarrhea. Staphylococcus aureus, causes skin infections, sepsis. Mycobacterium tuberculosis, causes tuberculosis. 2. Pathogenic viruses. Influenza virus, causes flu. Hepatitis B virus, causes liver disease. HIV, causes AIDS. 3. Pathogenic fungi. Candida albicans, causes mucosal fungal infections. Aspergillus, causes lung infections. 4. Pathogenic parasites. Plasmodium, causes malaria. Entamoeba histolytica, causes amoebiasis. C. Infection and immunity. 1. Infection, the process by which microorganisms invade the body and cause damage to tissues. Infections can be local, restricted to one part of the body, or systemic, spread throughout the body. 2. Immunity. The body's defense system involves white blood cells, antibodies, and other mechanisms to fight off pathogens. Immunity includes two types: innate, non-specific, and adaptive, specific immunity. 3. Clinical microbiology. A. Isolation and identification of pathogens. 1. Culturing methods. Using selective and differential media to grow pathogens from clinical samples like blood, urine, sputum, or stool. Example: McConkey agar for gram-negative bacteria. 2. Biochemical tests. Sugar fermentation tests, gas production tests, and enzyme tests like catalase and oxidase to help identify bacteria. 3. Serological tests. Using antibodies to detect the presence of pathogens or pathogen products in patient samples. 4. Molecular methods. Techniques like PCR, polymerase chain reaction, used to directly identify DNA or RNA from pathogens. B. Infectious diseases and diagnosis. 1. Respiratory tract diseases. Infections such as pneumonia caused by Streptococcus pneumoniae or influenza virus. 2. Gastrointestinal diseases. Infections caused by bacteria like Salmonella or Escherichia coli and parasites like Giardia lamblia. 3. Sexually transmitted diseases, STDs, diseases like gonorrhea, syphilis, and HIV transmitted through sexual contact. 4. Environmental microbiology. A. The role of microorganisms in the environment. 1. Organic degradation. Microorganisms help decompose organic matter like leaves, waste, and debris into substances that can be reused by plants and other organisms. 2. Biogeochemical cycles. Microorganisms play key roles in the nitrogen, carbon, and sulfur cycles, which are crucial for maintaining ecological balance. 3. Pollution and bioremediation. Using microorganisms to address pollution, such as employing bacteria to break down spilled oil, bioremediation. 5. Industrial microbiology. A microorganisms in industrial production. 1. Fermentation. Microorganisms are used to produce products like alcohol, acetic acid, and antibiotics through fermentation processes. 2. Probiotics. Beneficial microorganisms like lactobacillus used in health products such as yogurt. 3. Biopesticides. Microorganisms used to control agricultural pests in an environmentally friendly manner, such as using Bacillus thuringiensis. 6. Molecular microbiology. A PCR technology. Polymerase chain reaction. 1. Definition. PCR is a laboratory technique used to amplify, copy, specific DNA segments for further analysis. Used for pathogen identification or detecting genetic mutations. B. DNA sequencing. 1. Definition. Sequencing is the process of determining the nucleotide sequence of DNA. It is used to identify microorganisms or map their genetic makeup. C. Microbial genetics. 1. Horizontal gene transfer. The process by which microorganisms transfer genetic material between individuals, such as via conjugation, transduction, or transformation, which can lead to antibiotic resistance. 7. Antibiotic resistance and infection control. A mechanisms of antibiotic resistance. 1. Reduced membrane permeability. Bacteria alter their cell membrane structure so that antibiotics cannot enter the cell. 2. Active efflux. Bacteria use pumps to expel antibiotics that enter the cell. 3. Antibiotic destroying enzymes. Some bacteria produce enzymes like beta-lactamase that degrade antibiotics, such as penicillin. B. Infection control. 1. Prevention, vaccination, isolating infected patients, and maintaining good hygiene practices to prevent the spread of diseases. 2. Therapy, treatment with antibiotics, antivirals, or antifungals based on the identified pathogen. 8. Epidemiology of microbiology. A transmission of infectious diseases. 1. Transmission routes. Infectious diseases can be transmitted through direct contact, e.g., wounds or sexual contact. Airborne routes are contaminated food and water. B. Outbreak control. 1. Surveillance, monitoring disease spread to detect outbreaks early. 2. Quarantine, isolating infected individuals to prevent further spread of the disease.